Welcome back to day 2 of the Landscape of Cancer Research 2023. I'm Princess Senate, conference producer at Technology Networks, and I am here to introduce our first talk of the day, the exploitation of lysosomes in cancer therapy with graphene-based nanomaterials. I am pleased to welcome Dr. Ljubica Harhaji as your presenter. Dr. Ljubica Harhaji is a principal research fellow at the Institute for Biological Research in Serbia. Prior to this role, Dr. Harhaji was a principal investigator at the Ministry of Education, Science, and Technological Development of the Republic of Serbia, where she investigated and led the research on the role of autophagy in tumor cell death. She also received the L'Oreal UNESCO for Women in Science Awards in Paris, France, for the project The Role of Autophagy in Growth, Metastasis, and Chemotherapy in 2011. Dr. Harhaji completed her postdoctoral fellowship at the Panum Institute, University of Copenhagen, and received her PhD at the University of Belgrade, Serbia, where she also earned her bachelor's and master's degree. A warm welcome to you, Dr. Harhaji. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk. So feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Dr. Harhaji's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the video player. Finally, before we introduce our first session of day 2, we would love to hear your thoughts on the challenges researchers are currently facing. A brief poll should be popping up on your screen now. And the question is, in your opinion, what is the most pressing challenge in cancer research today? The options available for selection are A. Lack of funding for research, B. Limited access to innovative treatments, C. Understanding the underlying causes of different cancer types, and D. Addressing health disparities and improving access to care. I will give you a few seconds to respond. Thank you to all who responded. It is great to hear your opinions on this field. And if there are any other challenges you feel should be addressed, please mention them in the chat box now. And how do you think we can tackle these? With that out of the way, I will now hand over to Ljubica to begin her presentation. Degree. A warm welcome to you, Dr. Harhaji. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk. So feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Dr. Harhaji's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the video player. I will now hand over to Dr. Harhaji to begin her presentation.
Hello, well, my name is Ljubica Harhaji-Trajkovic, and today I am going to tell you something about how lysosome may be used in cancer therapy with graphene-based nanomaterials. Well, despite significant efforts and money invested in cancer research, cancer is still second leading cause of death. The main problems of cancer therapy are drug resistance and harmful side effects of drug therapy. Drug resistance is the consequence of uncontrolled cell proliferation and high mutation rate, which allow rapid selection of drug-resistant cell clones. On the other side, harmful side effects are the consequence of the fact that anti-cancer drugs usually interfere with signaling pathways that are common for both malignant and healthy cells. Therefore, we need something special in cancer cells which should be targeted. Lysosomes of cancer cells have some specific features. Let me first remind you what are lysosomes. Lysosomes are degradative acidic organelles. Their low pH of about 4.8 is maintained by the activity of vacuolar ATPase, the enzyme which pumps protons inside the lysosomal lumen. Lysosomes contain over 60 hydrolytic enzymes. Among them are proteases, nucleases, lipases, and glycosidases, etc., etc. Lysosomes degrade extracellular materials which are internalized in cells by caveoli-mediated endocytosis, Platri-mediated endocytosis, micropinocytosis, or phagocytosis. Lysosomes also degrade intracellular material which are obtained by autophagy. Lysosomes are necessary for tumor growth and metastasis. Autophagy removes damaged organelles and proteins which are accumulated during uncontrolled cell proliferation. Moreover, autophagy provides substrates for energy metabolism and building blocks which are necessary for de novo synthesis of micromolecules in fast proliferating tumor cells. Moreover, lysosomes enable invasion as they secrete catepsins and metalloproteases in extracellular matrix. Those enzymes digest extracellular matrix. Moreover, lysosomes secrete protons in extracellular space, which also stimulates invasion of cancer cells. Finally, lysosomes can trap and exocytose weak base hydrophobic antitumor drugs, which induce drug resistance. Therefore, it is not surprisingly that cancer cells have increased number and volume of lysosomes. Moreover, their lysosomes have increased concentration of hydrolytic enzymes. Because of that, because of their huge size and because of some specificities in the structure of their membrane, lysosomes of cancer cells are extremely fragile. Their membranes have increased concentration of destabilizing molecule, which is called sphingosine, while they have decreased concentration of stabilizing molecule, which is called LAMP. Moreover, lysosomes of cancer cells are full of iron, and iron catalyzes production of hydroxy radical, which is extremely toxic and may damage lysosomal membrane from inside. This induces lysosomal membrane permeabilization, or LMP for short, which may be followed by lysosomal cell death, or LCD. Graphene nanomaterials are made from graphene or graphene-like materials. They are made from one to several layers of carbon atoms, so they are high of about several nanometers, while their lateral dimensions are much larger and maybe up to several thousand nanometers. Their main representative is graphene. Graphene is planar monolayer of carbon atoms. They are arranged in two-dimensional honey-bomb lattice. Each carbon atom is connected with three neighboring carbon atoms with three sp2 sigma bonds, which gives graphene extreme mechanical strength. Moreover, there is one valence band which extends over the whole sheet of graphene, and it is responsible for high and unusual conductivity of heat and electrons of graphene. Unfortunately, graphene is hydrophobic and therefore it cannot be dissolved in water, and its synthesis is quite expensive. It can be synthesized by both bottom-up and top-down methods. Bottom-up means that it is synthesized from carbonaceous precursors, while top-down means that carbon materials with a lot of layers are transformed to material with single layer. Fortunately, the synthesis of graphene oxide is much easier and less expensive. Graphene oxide may be synthesized by oxidation and exfoliation of graphite. Graphene oxide contains oxygen-containing groups such as hydroxyl, peroxide, and carboxyl groups, They can be modified chemically, and they provide graphene oxide with water-soluble ability. Importantly, upon radiation with near-infrared radiation, graphene produced heat, and therefore it can be used in photothermal therapy. Graphene oxide is very biocompatible and it exhibits low toxicity. Reduced graphene oxide is produced by a reduction chemical, thermal or photothermal of graphene oxide. Because of that, it has less oxygen-containing groups, and it is similar to graphene. It is hydrophobic as graphene, but it can also produce heat upon radiation with the near-infrared light. Moreover, there are graphene quantum dots. Graphene quantum dots are smaller than previously mentioned graphene-based nanomaterials. Their lateral dimensions are less than 30 nanometers. They are called zero-dimensional materials. They have increased ratio between surface and volume. They are also synthesized by bottom-up and top-down synthesis. Bottom-up from benzene derivatives, and top-down they may be synthesized from graphene oxide and graphene as well. Importantly, they are soluble in water, they are non-toxic, biocompatible, and they have ability to fluoresce. Therefore, they can be used for bioimaging and biosensing. They also produce heat upon radiation with near-infrared light. But they can be also used in photodynamic therapy because they are able to produce reactive oxygen species upon radiation with visible light. So graphene-based nanomaterials have a large surface area, and because of that, they can be loaded by various anti-cancer drugs. Among them are doxorubicin, cisplatin, proflavin, and various anti-cancer agents, such as photosensitizers, microRNA inhibitors. Drug can be bound to graphene-based nanocarrier by pi-pi stacking, electrostatic and hydrophobic interactions. Those graphene-based nanomaterials increase drug solubility and bioavailability. They also protect drugs from degradation and premature release. Moreover, if ligands for folate, transferrin, and epidermal growth factor receptors are attached to graphene nanocarrier, they increase their therapeutic efficiency and decrease side effects because receptors for these ligands are overexpressed on tumor cells. These graphene-based nanocarriers usually are connected with endosomolytic agents. Among them are polymers, pH-sensitive peptides, and proteins, which enable anti-cancer drug to escape from endosomes and lysosomes. Now I will tell you how graphene nanomaterials arrive in lysosomes. First, I will explain you what is enhanced permeability and retention effect, or EPR effect for short. Well, blood vessels differ in tumor tissue from normal tissue because they have their endothelium possess large pores. It is irregular. They lack smooth muscle layer and usually don't have lymphatic drainage. So nanomaterials, which are nano-sized, such as graphene nanomaterials, easily escape from endothelium of tumor tissue and goes in tumor tissue, and then they retain there because there is no lymphatic drainage. So graphene nanomaterials will preferentially accumulate in tumor tissue. Thereafter, they are internalized in tumor cells, and this can be done by several mechanisms. First one is clathrin-mediated endocytosis, which is characteristic for small graphene oxide particles with diameter less than 1400 nanometers, for graphene quantum dots and reduced graphene oxide. It is facilitated by the surface receptors and begins with the formation of clathrin-coated vesicles. It matures in early endosomes with pH of about 6.5. These early endosomes mature in late endosomes with pH of about 5.5, which then fuse with lysosome and form endolysosomes with pH of about 4.5. Moreover, there is a phagocytosis, which is characteristic for large graphene oxide particles with diameters between 6 and 800 nanometers. These nanoparticles are perceived as foreign organisms, and they are internalized in organelles which are called phagosomes. Phagosomes then also fuse with lysosome and form phagolysosomes. Small hydrophobic graphene may directly penetrate into tumor cells through the plasma membrane, and in cytoplasm it can be scavenged by phagophora, which form autophagosomes, which can also fuse with lysosome and form autophagolysosomes. Finally, some graphene quantum dots may be internalized by caveoli-mediated endocytosis, which begins at lipid rafts. First, caveoli vesicles is formed. These caveoli vesicles usually don't fuse with lysosomes, but with endoplasmic reticulum and Golgi, but it may also fuse with endosomes to form caveosomes, which can finally fuse with lysosome. So the question arises, can those pure graphene-based nanoparticles be degraded in lysosomes? Well, there are conflicting results. It is demonstrated that macrophages can degrade graphene, but the mechanism is unknown. Moreover, it is demonstrated that graphene can be degraded in extracellular space by lysosomal enzymes. On the other hand, graphene oxide cannot be degraded in tumor cells. It accumulates in tumor cells and makes steric hydrons. So if anti-cancer drug is connected with graphene-based nanocarrier, it should escape from endosomes and lysosomes if it acts on organelles other than lysosomes, for example, nucleus or mitochondria on endoplasmic reticulum, etc. So how nanoparticles escape from lysosomes? There are several mechanisms. The first one is proton sponge effect. It says when we have molecules with amine groups and buffering capacities, such as PEI or PAMAM, they are able to absorb protons. Therefore, vacuolar ATPase try to maintain lysosomal pH and pumps protons inside the lysosomal lumen. This induces chloride ions to enter the lysosomes, as well as diffusion of water molecules, and because of that, lysosome burst because of increased pressure inside. There is also the umbrella effect. It is a kind of extension of proton sponge effect. It says that if we have protonation of amines, it will become nanoparticle will become positively charged, and there will be a charge repulsion. Nanoparticle will extend, and it will induce lysosomal rupture. Moreover, in the pore formation mechanism, nanoparticles directly fuse with lysosomal membrane, induce its tension and stress, and there is formation of pore which enable anti-tumor drug to be released from lysosomes. And at the end, there is a mechanism of photochemical destruction. In this mechanism, photosensitizer, which is connected with graphene nanomaterials, produce reactive oxygen species upon radiation with visible light, and these reactive oxygen species are able to induce lysosomal rupture. But it is not necessary that entire graphene-based nanocomplex exit from lysosomes. It is enough that only anti-cancer drug exit from lysosomes to fulfill its anti-cancer activity. So in that case, anti-cancer drug should be first separated for graphene nanomaterials, and it can be done by several mechanisms. The first is protonation of both graphene nanomaterials and drug, which become positively charged, and there is electrostatic repulsion between them. Moreover, low pH can induce breakage of pi-pi interactions, which are formed between graphene nanomaterials and anti-tumor drugs which possess aromatic rings, such as doxorubicin, proflavin, chlorine, E6. Moreover, low pH may induce breakage of amide and carboxylate bonds, but pH as well as high concentration of reactive oxygen species inside the lysosomes may induce breakage of deselenide bonds. So let us see some concrete examples from scientific literature with graphene nanomaterials and lysosomal escape. As you can see on the left side, nanoparticles from about 20 nanometers to about 1050 nanometers can all be internalized in tumor cells and may escape from them, from tumor cell lysosomes. They are, with one exception, they are usually negatively charged. Those are colored in red, but they become positively charged inside the lysosomal lumen because they are protonated. And the consequence of this is repulsion between anti-cancer drug and the rest of the complex. So they are separated, and these examples are colored in purple. Moreover, low pH may induce hydrolysis of amide links, breakdown of pi-pi interactions, cleavage of carboxylate bonds. If photosensitizer, for example, androcean and green, are part of the complex, upon radiation with near-infrared radiation, they induce production of reactive oxygen species and cleavage of deselenide bond. When drugs are separated from the nanocarrier, then they escaped from a lysosome mainly by proton sponge mechanism, which is enabled by the presence of PAMAM in the complex, but they can also escape by the photochemical destruction, which is produced by photosensitizers irradiated with light, which produce reactive oxygen species, or by heat, which also damages lysosomal membrane, and this heat may be produced by both irradiated photosensitizers and reactive and nanomaterials. Well, next, escape of anti-tumor drugs from lysosomes may be associated with lysosomal membrane permeabilization, which may induce leakage of lysosomal enzymes inside cytoplasm. It is important to say that cathepsin B, D, and L retain the activity at neutral pH of cytoplasm, so they degrade cytoplasmic proteins, and this may have as a consequence, lysosomal cell death. Lysosomal cell death may have both apoptotic or necrotic feature. If there is limited LMP, it will have apoptotic feature, but extensive LMP will induce necrosis. So all previously mentioned mechanisms of lysosomal escape may be followed by lysosomal cell death. However, if there is dominance of some other kind of anti-cancer mechanism, lysosomal cell death may not have time to occur. Moreover, if the drugs escaped from through pores, these pores may be large enough to let anti-cancer drug to escape from lysosomes, but not large enough to let catepsins to exit from lysosomes, because catepsins are extremely large molecules. Finally, we should make distinction between escape from lysosomes and late endosomes, because lysosomes are more acidic and they have mature enzymes in contrast to late endosomes. So lysosomal cell death is more likely to occur when drugs escaped from mature lysosomes than endosomes. The main inducer of LMP in all the studies with graphene nanomaterials are reactive oxygen species. Importantly, because of fast metabolism, tumor cells have increased breakdown of proteins which contain iron. This iron is able to catalyze Fenton reaction, in which hydroxyl radical is produced. This hydroxyl radical is extremely toxic and it can damage lysosomal membrane from inside. Importantly, lysosomes lack antioxidative defense. They do not have catalases, superoxidismutases, and glutathione peroxidases. So let us see some examples from the literature. In the first example, graphene nanosheets reduced mitochondrial electron transport activity. This induced synthesis of reactive oxygen species, which then induced lysosomal membrane permeabilization. In the third example, light irradiated photosensitizer, fluorine E6 or ruthenium, also produced reactive oxygen species, which then induced catepsin release in cytoplasm. And in the fourth example, low pH of lysosomes induce a release of iron from transferrin, which is part of the complex, and released iron together with antimalaric dihydroartemisinin, which is also part of the complex, produce reactive oxygen species, which then induce lysosomal damage. Moreover, lysosomal damage may be also mechanically induced by sharp edges and round surface of graphene nanosheets. And there is one digression in this study. Separation of anti-cancer agent from nanocarrier is made not by low lysosomal pH, but by activity of lysosomal enzyme catepsin B. So let me tell you something about autophagy. Autophagy is process of degradation of damaged cytoplasmic macromolecules and organelles in organelles which are called autophagolysosomes. It begins with sequestration of intracellular material in phagophore. This phagophore mature in the autophagosome. Autophagosomes is then fused with lysosomes in organelle which is called autolysosomes, and their intracellular material is degraded. So the question arises, if graphene nanomaterials are externally added, how they end up in autophagy, which is characteristic for intracellular materials. First of all, I will remind you that hydrophobic small graphene nanoparticles may directly penetrate through plasma membrane in cytoplasm and then can be part of autophagic process. Alternatively, if the graphene nanomaterials are endocytosed in endosomes, then they can escape from endosomes into cytoplasm, and then they can be scavenged by phagophora. Or endosomes may fuse with autophagosomes, and this will prograde in the process of autophagy. The role of autophagy is primarily protective. Autophagy protects cells from accumulation of damaged proteins and organelles, from energy deficiency, from oxidative or drug-induced stress, and this is called protective autophagy. So if we see other examples from scientific literature, we can see that there is cytoprotective autophagy, which protects cells from graphene nanomaterials. However, this protective autophagy may be suppressed. It means it is not completed, it is induced, but at the end, autophagy degradation, which is called autophagy flux, is suppressed. So this suppressed autophagy will lead to decrease of cell viability. It will be cytotoxic. There are 4 examples in literature. In the first one, non-degraded graphene oxide induced steric hydrons in the lysosomes. It will increase lysosomal pH, which will decrease activity of lysosomal enzyme. The consequence will be accumulation of dysfunctional mitochondria and mitochondrial apoptosis. Moreover, in the second and third example, LMP, which is induced by graphene nanomaterials, which will increase lysosomal pH, which will reduce activity of lysosomal enzymes and induce accumulation of lysosomal substrate of autophagic proteolysis, which is called protein 6-2, and this will induce apoptosis. Moreover, accumulation of undigested substrates may also induce necroptosis. Moreover, autophagy may be also cytotoxic. For example, if it is unregulated and extensive, it may digest functional mitochondria or some pro-survival molecules. So it may induce cell death by necrosis, apoptosis, necroptosis, etc., or it can be a mechanism of cell death itself, and it is then called programmed cell death type 2. So we also have examples when graphene nanomaterials induced such cytotoxic apoptosis. Lysosomes are not only involved in anti-cancer effects of graphene nanomaterials, but they can only help us to detect cancer cells. Graphene nanomaterials have ability to quench fluorescence of fluorescent dyes. It is shown that reduced graphene oxide or graphene itself may reduce fluorescence of BODIPOI, chlorine E6, or indocyanin green. But when these nanocomplex arrive in tumor tissue due to EPR effect and are, for example, endocytosed, and after endosomes, they end up in endolysosomes with low pH. These fluorescent agents are detached from graphene nanomaterials and they start to fluoresce. So on this way, we can detect tumor cells, but may also know when the drug has entered tumor cells. So let me say something about future perspectives. As you can see from this lecture, The most studies dealing with lysosome graphene nanomaterials were demonstrated how graphene nanomaterials separates from antitumor drugs in lysosomes and how then antitumor drugs escape from lysosomes. And most of them were shown that physiochemical changes of drugs and graphene nanomaterials which occur due to low lysosomal pH are responsible from separation of drugs and nanocarriers. There is only one single study which demonstrated that activity of lysosomal enzyme is responsible for separation. So this possibility should be further investigated. Moreover, a lot of studies are investigating the role of autophagy in antitumor cells induced by graphene nanomaterials, but there are only a few studies which demonstrated the ability of graphene nanomaterials to induce lysosomal cell death. So having in mind that lysosomes of tumor cells are extremely labile, that they possess a huge concentration of iron, and we suggest that this possibility should be further investigated, especially because cells which are resistant to apoptosis may be killed by lysosomal cell death. This ability of graphene nanomaterials to induce lysosomal membrane permeabilization and lysosomal cell death may be increased by some chemical modification, for example, by functionalization with amine-modified polystyrene, as has been done for some other types of nanoparticles. Moreover, graphene nanomaterials may be loaded with antitumor drugs. They can induce lysosomal membrane permeabilization themselves, such as some inhibitors of vacuolar ATPase, cationic amphiphilic drugs. And in the end, I want to tell you that the graphene quantum dots are detected inside the lysosomes and that they are able to produce reactive oxygen species upon radiation with visible light. Moreover, graphene quantum dots are detected inside the lysosomes and they're able to produce reactive oxygen species upon radiation with visible light. So maybe they are able to produce lysosomal membrane permeabilization and lysosomal cell death, which should be investigated. At the end, I want to tell you that before all of this, we should investigate the safety of usage of graphene nanomaterials in cancer therapy. And in the end, I would like to thank you for your attention. If you're interested in more details about today's lecture, you can read it in our review article, which is still as a preprint version. And at the end, I would like to ask you if you have some questions.
Thank you very much, Ljubica. That was a fantastic presentation and a great way to kick off the second day of the event. As with all the talks, we are likely going to have lots of questions from the audience. I can see we have received some questions already, so I'll move straight on to the Q&A now. For the first question, are there anti-cancer drugs that are inducers of lysosomal membrane permeabilization?
Yes, there are some. There are, for example, destabilizer of macrotubules, for example, paclitaxel, vincristine, and docetaxel. There are also some antiproliferative drugs, such as fludarabine and cisplatin, inhibitors of BCL2, for example, obatoclax, ubiquitin proteasome inhibitor, botrezomib, and two inhibitors of tyrosine kinase, imatinib and sorafenib. Moreover, there are a lot of LNP-inducing drugs which are used to treat some other diseases. Among them are various antidepressants, antibiotics, immunosuppressants, and antimalarials, and this drug can be repurposed for cancer treatment as well.
Thank you, Ljubica. Moving on to the next question, could you summarize which classes of agents have been linked to GNMs in testing their anti-cancer activity?
Well, those are, first of all, some anti-cancer drugs, for example, doxorubicin, cisplatin, roflavin. So various other anti-cancer agents, for example, photosensitizers, BODIPY, and chlorine E6, as well as indocyanine green. Then some inhibitors of bronchogenic microRNAs. Graphene nanomaterials can also functionalize with specific antibodies or ligands for folate transferring epidermal growth factors that are overexpressed on cancer cells. And of course, with endosomolytic agents such as pH-sensitive peptides, various polymers and proteins, for example, PEI or PAMAM.
The information you provided was both excellent and informative. The next question I have for you is, are there clinical studies with GNMs?
Actually, not much. Graphene nanomaterials have been tested for external use, for example, as material for condoms, for dental implants, as stickers to relieve pain from menstrual pain, and sensors for heart attack. But there is one study which is still in the recruitment phase, which plans to investigate ability of graphene nanomaterials to be used as photothermal adjuvant in COVID therapy. But it is still not finished. I'm not sure if it has begun. There is also a study which investigates adverse effects of graphene inhalation on the cardiovascular system, but no results were posted. Therefore, there are not much data on safety of graphene nanomaterials for clinical applications. But on the other hand, there are a lot of animal studies. Unfortunately, they showed that graphene are retained in lung, liver, spleen, and bone marrow, and they can induce acute inflammation and chronic injury to important organs. They also may have hemolytic activity, and they may damage embryogenesis in zebrafish. However, the toxicity of GNMs mainly depends on their size, type, functionalization, concentration, and entering route. But there are some. Oh, yeah.
Oh, sorry. Thank you, Ljubica. It looks like we have some more time to answer another question. Is it better for cancer therapy if GNMs accumulate in lysosomes or escape from them?
Well, it depends. A lysosomal accumulation may suppress autophagy flux. So if autophagy is protective, its inhibition will stimulate tumor cell death, and it will be good. On the other hand, lysosomal accumulation is bad for anti-tumor drugs which act on organelles other than lysosomes, for example, nucleus, mitochondria, and the plasma reticulum. Moreover, these drugs should avoid degradation in lysosomes, so they should escape from lysosomes. And finally, lysosomal escape may be beneficial because it can be followed by lysosomal cell death.
It looks like we have some time to answer one final question. Why is it important to monitor the entry of GNMs into lysosomes?
Well, after preferential penetration into tumor tissue, graphene nanomaterials usually end up in lysosome tumor cells, which allow us to detect tumor cells. Moreover, if photosensitizer is, for example, bound to graphene nanomaterials, by monitoring its entry into lysosomes, we can determine appropriate time and location for light irradiation in photodynamic therapy.
Thank you so much, Ljubica. Unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Ljubica, and we'll be back soon with the next talk.
Thank you.